Okay. Okay. So this is a review session for the Chemistry of Life test that you guys are going to be taking tomorrow and the next day. And 3 o'clock on Wednesday, and here we are. And so I kind of had some ideas on what you might need to know, but why don't I open it up to you guys first? If some of you really have some specific questions, I can address those first. Or would you like me to just plow into it myself? That's fine. Oh, structures, yes. Okay, well, you know what? Probably the best way to do that is to, let's go to that pattern matching that I assigned to you, because I think that's a good place to kind of look at structures. So let me go to, because I have it open now. So again, once I share the screen, I won't be able to see your chat. So if you have questions, you'll just have to open your mic up and talk to me. Okay. Okay, so let's accordion that. And let's go to, where did I have it? I think it's right. Here it is. Okay. All right, so can you see this? Need somebody
yes.
to.
We can see
Okay.
it.
Thank you. Okay. So I thought, why don't we go up to this one first, because this one has kind of has a smattering of each type of thing. Now, when you looked at those, at these shapes, right? Like if you look at something like this that has all the carbons and hydrogens and oxygens on it, all of these structures that include every carbon, hydrogen, oxygen, nitrogen, and so forth, these structures are usually monomers. They're the smaller building blocks of the larger molecules that we're going to talk about a lot. And it is true that some of the carbohydrates just exist as small monomers. For example, this is glucose. And one of the clues for telling which monomer is which is I kind of open these up and you know, I'll show this one over here. Here's a different one, and you can see that those are different structures. And again, both of these are monomers. They're just monomers of different things. The reason that I know that this is a carbohydrate is because it has carbon, hydrogen, and oxygen in it all alone. And it has the ratio of 1 to 2 to 1, carbon, hydrogen, oxygen. It has a lot of oxygen in it. There's one there, there's one here, there's one here. So if you have a molecule that is showing you carbon, hydrogen, oxygen only, and has a lot of oxygen, that's going to be a carb. Again, I'm sorry, I can't see your chat, so you have to ask me out loud. Do you have a question?
Yeah, sorry, I was just trying to raise my hand. I was wondering, I saw something about a phosphoated ester bond. Do you know what that is?
Phosphodiester bond. I
Yeah.
can go over that in just a little bit, but right now I'm just going to start with some basics, okay?
Okay, yeah, sorry.
Is it Reese? I think that's you, right?
Yeah.
Okay, so yeah, that's probably a good one to go over. I'll go over that when I talk about types of bonds, which I will talk about, okay? But this, I can tell this is a carbohydrate, again, because it has carbon, hydrogen, oxygen, and lots of oxygen. So that's going to be a carbohydrate, and this one is glucose. So if I were classifying it, if I were classifying it, you know, I would put it together with other carbohydrates. And in fact, I already have this one below it is also a carbohydrate, and I can tell because of the ratio of the atoms. And I can also tell that in this case, we don't have a monomer. We have 2 monomers that are bonded together, and this happens to be a disaccharide, which is what we call a carbohydrate when it's made of 2 monomers. Okay. So 2 glucoses together, and a glycosidic linkage, which is one of those bonds that Reese was asking about, right? So the glycosidic linkages are the bonds that are between carbohydrate monomers, monosaccharides. I'm going to put that back, and that goes, that's a good structure to be able to recognize, right? So here's a monosaccharide, glucose, and here's 2 of them bonded together right there. That's how you can tell carbohydrate.
Are glycosidic linkages covalent?
Yes, very good question. The other thing that I wanted to make sure you understood is that really the only bonds that we're talking about mostly are going to be covalent bonds and hydrogen bonds, right? Those are two very different bonds in nature. All covalent bonds share electrons and are pretty strong, and you find them within molecules, holding atoms together inside molecules. But there are different types of covalent bonds that we've looked at more specifically. So a glycosidic linkage is just a type of set of covalent bonds that exists within a polysaccharide chain, right? Peptide bonds are a type of covalent bond that links amino acids together. Still covalent, but we call it peptide bond as a specific thing that's only found in proteins. I'll just keep going with this. So if you find what was the other one? A phosphodiester bond linkage or bond is found in DNA molecules because you have phosphate in DNA, and that's what links nucleotides to each other. So that answers your question, Reese. And then the one that links fats together are ester linkages. But all of the things I've just mentioned, they're all covalent. They just have special names because of the type of molecules they are found in. Okay, so then the other category of bonds is hydrogen bonds, which are much, much, much weaker than covalent bonds. And the main one we have seen so far is hydrogen bonding that exists between water molecules, right, which we talked about a lot, which will be on the test, right? And hydrogen bonding between the positive ends of one water molecule and the negative end of the other, it's kind of a weak attraction. But if you put it in a billion water molecules in a glass of water, those intermolecular forces, hydrogen bonds, result in cohesiveness and adhesiveness and all those other things, right? So there's a bit about bonding. All right, so the carbs are here, I put them together. This, I can tell, is not a carb. And the reason I can tell it's not a carb, let me make it even bigger, is if you look at the atoms in this, you will find that there is a nitrogen there and that there is a sulfur there, right? So this can't be a carbohydrate, must be something else because it's got carbon, hydrogen, oxygen, and nitrogen and sulfur. So immediately when you see the sulfur, you should think, that's in proteins. Sulfur is always in proteins as far as elements go. So this is not a protein. This is a monomer of a protein called an amino acid. Particular amino acid, and all amino acids have this in common. They all have the central carbon with a carbon OOH carboxylic acid group over here and an H3 amino group over here. All amino acids have this in common. So that's what you would look for if you were looking for an amino acid structure. And then this down here is the R group that makes one amino acid different from another amino acid. So this is an example of an amino acid that if you bind it together with lots of peptide bonds, you get a polypeptide, and eventually you get a protein if you fold it properly into the right three-dimensional shape. So this is amino acid. So I'll put all the amino acids in one place here. I'll make them small so they fit there. So there's an amino acid. I think this also is one. Take a look at this. Yeah, you see that? So hopefully you can see that nice and big. There's that central carbon, there's that carboxylic acid group again, there's the NH3 amino group again. All amino acids have this in common. The R is slightly different, so this is a different amino acid from that other one I showed you, but it's still a monomer of proteins. So I can put that into the same category as the other amino acids. They're both amino acids. They contain sulfur. They contain an amino group and R groups that make them different. Okay, so then in this category here, take a look at this. Make it nice and big. Oh, ignore that. There. Okay, so this right here is a really, really important molecule. Can somebody tell me what they think it is?
Phospholipid.
Yes, it's a phospholipid. Yes, so it goes in the lipid group. So it's a weird lipid. It's a bit weird because it looks like it almost could be a fat, except that a fat would have 3 of these long chains, 3 of them, and a phospholipid only has 2. And then the other thing the phospholipid has is a phosphate group, hence phospholipid. And that makes phospholipids also pretty weird because generally lipid molecules do not have any phosphorus in them. Lipid molecules are as in terms of their elements, are very similar to carbohydrates. They have carbon, hydrogen, oxygen, but if you look, you find that you have much less oxygen in a lipid molecule. Lots of carbons and hydrogens, tons of them, which makes them nonpolar because carbon and hydrogen have a hydrophobic quality to them. But only a few oxygens. There's your ester linkage, by the way, right there. So there's an ester linkage that links a fatty acid to a glycerol. And there's another one right there. There's two linkages. And then there's a phosphate here. Okay, function of the phospholipid, really, really important to know. Where do you find these things?
Cell membrane.
Cell membranes, excellent. Okay, so you may remember the phospholipid bilayer of the cell membrane. Most of a cell membrane is made up of these guys, so that's important. Okay, and I think you also learned, hopefully you learned amongst your travels and in your mastery quizzes, that this end over here is not hydrophobic. It's kind of an exception to the hydrophobic rule of all lipids being nonpolar and hydrophobic. But this particular bit of a phospholipid is actually polar and hydrophilic. It likes water. So this will be the part of the molecule that is in contact with the water outside the cell, and over here, if there was another one over here, it would be in contact with the water inside the cell. And it makes the lipids naturally line up together into that lipid layer so that the hydrophobic parts are pointing in because they don't like the water. They point away from the water, right? These long hydrophobic parts mean that most of the molecule is still, like most lipids, doesn't like water, hydrophobic. But the hydrophilic end here means that this molecule makes a very good cell membrane because they naturally line up. So these ends are near the water on both sides, where the cytoplasm is and where the outside of the cell is. And these hydrophobic parts make a really nice barrier that prevents most molecules from getting in unless the cell needs it there. So there's your phospholipid. That's good to know. Okay. And I can put that over here. I'm going to put fats in this thing here. And this thing, what is this? Let's open that up because that goes with phospho. Oops. Okay, open it up a little bit. All right. Okay, so this is sort of, it's not really a monomer, but you could sort of call it a building block of fat molecules. This is a fatty acid. And this particular fatty acid, you know, if you look back at the phospholipid, and I enlarge that, it's kind of like this fatty acid, if you turned it on its side, it would be this piece right here. So a fatty acid like this one makes up fat molecules or phospholipid molecules. It could be in there as well. And there's different types of fatty acids. You notice that this fatty acid is very straight. And I talked about that. I talked about the fact that, you know, when you have straight molecules, they tend to pack much more tightly. Whereas when you have bent molecules, like this fatty acid has a double bond in it right there, and that causes a kink in the chain. That would be an unsaturated fatty acid and would be more likely to be found in an oil rather than a fat that makes something solid like your solid fat or like butter, animal fat. Okay, so that's a little bit about lipids. There's no steroid here, but I'm not going to worry about that right now. So this goes, oh, make it small. Small. I'm just going to put it over there. Okay, so now the last group has these things in it. I'm just going to pull them all over here. Okay, so this group here was amino acids. Remember we talked about those, right? This group here was carbohydrates. So I had the glucose and I had the disaccharide there. And then this group over here was lipids. So we had the phospholipid and we had the fatty acid. This group here is the nucleic acid group. So the bits in here are all building blocks of large nucleic acids like DNA and RNA. And the building block, the monomer for nucleic acids is a nucleotide, and that's this one right here. So let me bring this up here. Bring it over here. Okay, so this is a nucleotide right here. Let's take a look at that and see how that is different from the other types of organic molecules. So with a nucleotide, oh, it doesn't show all the parts of it. Maybe this one does? Okay, no, so it doesn't. It must have gotten cut off. It looks like it got cut off. Okay, so what I was trying to show you is that, again, you have carbon, hydrogen, and nitrogen in this, right? Carbon, hydrogen, oxygen, and nitrogen, so CHON. And that might make you think that, well, is that a protein then? But it's not, because you're also supposed to have a phosphate group here, and I just don't see it. It doesn't look like it's here. So it may have gotten cut off. Oh, well, maybe I'll find another one. All right, so in a nucleotide, there are three things. There's a ribose, which is here. There's a base, could be A, T, C, or G, or U if it's RNA. And this is an example of a nucleotide base or nitrogenous base. And you can tell that this is not a disaccharide because you've got nitrogens in there. And there's no nitrogen in carbohydrates, so that should tell you immediately you're dealing with something else. Okay, so this is a part of a nucleotide. You've got two of the parts, but you're missing the phosphate. And it's the same with this one. If I bring this one over, it looks like that you've got two of the parts, but you're missing the phosphate. And this here, I'll make this nice and big so you can see it. This is one of the parts of a nucleotide also. This is another nitrogenous base, a pyrimidine, which is a single ring, right? So this is, it could be cytosine or guanine, whereas this could be guanine or adenine, cytosine or thymine, one of those. And you can see the nitrogens in it that tell you, okay, it's not a carbohydrate and it's not a lipid. All right, I'm going to go to another screen now. Okay, so let's talk about proteins for a minute, because proteins, I think, may be one of the more difficult concepts of you to get. So let me share something I just pulled up earlier. Let me show you this. Can you see that?
Yes.
Okay, great, thank you. Okay, so this looks pretty chaotic, I'm betting. But what you're looking at is a string of amino acids. You're looking at the primary structure of a protein. So you can, if you can kind of in your mind follow me, can you see where one amino acid begins and another ends? If you look at the screen, can you trace central carbon here, carboxylic acid, amino group? Here's one amino acid. Here's the next one. Here's another amino acid with a different R group sticking down. So you've got your amino group that's already formed a peptide bond with this amino acid here. So there's another amino acid. Here's another peptide bond here between this third amino acid right there. Each amino acid has a COO end and an NH2 end when it's by itself, when it's just dissolved in water. But when it's bound to other amino acids, it looks more like this. So there's another peptide bond there. Here's another amino acid with another different R group hanging down there. Here's another amino acid. Again, I can't see your chat, so you have to say it out loud. I'm sorry. There's another one. Very big R group on this one. Here's another amino acid, which just has a hydrogen atom for an R group. That's what's going on there. And this amino acid is the same. Glycine, there's glycine, is the same as the first one. If you look back here, here's glycine here, and also has an R group that's just a hydrogen atom, right? And then aspartame has an R group with an ionic end to it. There's another amino acid there. So this is a polypeptide chain here. It's a little short one, but it's showing you how you can take amino acids and string them together, right? So when you are putting two amino acids together, let's pretend that you are putting these two amino acids together. If you put them together and you're forming this peptide bond, what kind of a reaction is that? Putting things together, forming a new bond, losing water.
Dehydration synthesis.
You got it, exactly. Okay, so in order to link all of these amino acids up and create those bonds in the first place, the cell had to do multiple dehydration synthesis reactions. And every time they do that, you produce some water from that, every time you make a new peptide bond between amino acids. So this is the primary structure, or the beginnings of a primary structure of a protein. most proteins are made of more than just, let's see, eight amino acids. Most of them are longer than that. Okay, so hopefully that helps you kind of get into your mind how, you know, how the structures that were on that pattern matching page, what they have to do with the longer structure of the protein. So now let me go back to you. Okay, let me just come back here for a minute. Okay. Okay, so those are some basics there. I'm going to go through the levels of protein structure. Everyone okay with that?
Yeah.
Yeah. Okay. I'm assuming that that would be an area where you would need some help. So let's just do that here. All right. So primary level. Okay. Oh, I just spelled it wrong. The primary structure is just the sequence of amino acids. Just like I just showed. So literally you have a string of beads. That's what it is. Okay. And each little amino acid, there are 20 different kinds of them, might have slightly different R groups, and therefore will eventually affect the folding that comes next in different ways. And that can be very long. It can be up to 300, 400 amino acids in a chain. And each amino acid is bound to the next by a peptide bond. And so the whole thing then is called polypeptide. And that primary structure of those 20 amino acids, that's fodder for quite a diverse number of different polypeptide chains that form all the proteins in nature, in organisms, in all of the life that's on the planet. There are a billion different ways to combine 20 different things into a 500 thing chain. When you take into account some things are repeated, some things are over here, some things are over there. If you have 20 different kinds, you can make lots of different chains. And the thing that you need to realize is that this has a direct connection with the sequence of nucleotide bases in the DNA strand. So it's the information in the DNA that determines which amino acid comes first, second, third in that long chain of amino acids. So remember that DNA and proteins are very much connected. DNA gives the information that allows the cell to put amino acids into a certain sequence that then results in the structures that follow. So now let's go secondary structure. So the secondary structure can be one of two things. You can take that string of beads and you can twist it into a helix, or you can do a beta pleated sheet. Or okay. Either way, so your eyes are taking that string of beads, and I can't really draw an alpha helix. I'm doing the best I can there. You're either making an alpha twist, or you're making something that looks kind of like a folded piece of paper, up and down, up and down, up and down, right? And it all depends on hydrogen bonding between relatively adjacent to each other R groups. It depends on how the groups are interacting and forming that secondary architecture and holding those things in place, right? I'm drawing hydrogen bonding here between, I'm not very good at art, but you get the idea, okay? So hydrogen bonding between relatively close R groups. That's what holds secondary structure together, right? Tertiary structure is probably the most important. Tertiary structure is the 3D folding of if you have something that's twisted in an alpha helix or beta sheeted, you take that and you fold it into a globule or into something fibrous that has real three-dimensional architecture. And this depends on lots of different things. So if you're taking the secondary structure and then folding that again into something that depends on more distant R groups and also on the polar and nonpolar nature of the R groups. So, okay, so like this. So this is taking this and making a big thing about it. Okay, so this depends on several different things. This is bonding between more distant R groups. And that's going to include things like disulfide bridges. Disulfide bridges. Disulfide bridges are if you had a cysteine. Oh, geez, stop that. Okay, if you had a cysteine amino acid here, which has a sulfur sticking off of it, right? And then you had another one over here, which has another sulfur sticking off of it. They like each other, sulfurs. So they form a very strong covalent bond. They're another example of a covalent bond. But so disulfide bridges are very strong connections between distant groups that might really hold this loop very close to that one. There. That's only one example, though. There are others. There's also ionic bonds that can happen, where if you have an ionic natured R group coming off and meeting up with an opposite one. So there's ionic bonding, which is also very strong. Ionic bonds between distant R groups. And you can also have hydrogen bonding in here as well. There's all kinds of things going on between distant R groups. And then there's another thing that's important for you to know, called hydrophobic interactions. Okay. Hydrophobic interactions, now you have to kind of imagine in the line of amino acids up here, I'm going to use there we go. In the line of amino acids up here, you know that some R groups are nonpolar. They don't like water. Any of the nonpolar R groups that are sticking off on a line, those amino acids are going to tuck themselves in this area, in the center of the protein, away from the water. And that causes a lot of twisting and turning and arranging of that three-dimensional structure, which winds up being important for whatever protein you're talking about. So hydrophobic interactions are a big deal. If you have nonpolar R groups are going to be located inside, along the R groups inside the molecule. They're going to be in here. The hydrophilic, water-liking R groups are probably going to be like this out here. There's going to be hydrophilic amino acids there, and out here on the edges here is where you're going to have your hydrophilic R groups, the amino acids that have those hydrophilic R groups. Okay, how's things so far? Not too overwhelming? Maybe, maybe not. Maybe you'll listen to it later a second time before the test and it'll all come together for you. Okay, the final type of structure doesn't always exist. There are some proteins that don't have a quaternary structure. Quaternary structure. A quaternary structure just means that you have more than one blob that is now stuck together into a bigger blob. That's all it means. So quaternary structure means, and I have learned recently that it's hydrogen bonding that holds these guys together, is more than one polypeptide that's tertiarily folded and then stuck together to form a larger protein. Okay, so hemoglobin is a really good example of this. Hemoglobin is like, if you have one, I'll try to draw this again, I probably can't, but there. So this guy here is now times 4. I'm not going to do that very well. All right, so just, yeah, that looks really great, Mrs. Murdoch. There we go. So we have, instead of one polypeptide that's all the way folded to its tertiary level, you have 4 polypeptides that are all folded into their tertiary level and now are coming together to attach to each other to form a nice big molecule. And hemoglobin is a good example of that. Okay, actually covered quite a lot there. Covered a lot of what I thought would happen. Okay, so since we're on the subject of proteins, what's denaturation? Can you tell me what it is? Yes, Reese, go ahead.
The misfolding of proteins.
The misfolding of proteins. That's pretty close. I think I would just add a little bit more to it, though. What generally causes it?
Different pH levels, temperatures, and exposure to chemicals.
Yes, very good. So extreme pH, like a really strong acid, will do it. Extreme temperatures, like a really, really boiling hot water or environment, will do it. And what's really happening is the DNA's quaternary and tertiary structures are going to be the things most affected. So the folding will come unraveled. So when you're denaturing a protein, you're likely to affect the tertiary quaternary structures. It's all just going to come unraveled. And that is a problem for the function of the protein. The primary structure is almost never affected by a denaturation process. The peptide bonds are not going to be broken. It's more the hydrogen bonding and the hydrophobic interactions that come unraveled, right? So anything that's not a covalent bond kind of lets go, and that creates a different shape to the protein. And if the protein doesn't have the right shape, it will not work as an enzyme because the active site will not be the right shape to meet with its substrate. It won't work as a receptor molecule because, again, you need the right receiving area. It just won't work in terms of whatever it is supposed to do. So that's how denaturation works. It is affecting folding. It will not affect primary structure, though. What if they tell you, though, that there is a particular protein that had an amino acid removed? Then how will removing, like what if you removed this one? This one was out of there. Would that affect the next levels of structure? What do you think? Yes or no? It's very possible that it would. It just depends on where the removal happened. It will definitely affect the primary structure, and it's very possible that you also might affect the tertiary structure. In the case of hemoglobin, a prime example is that there is a mutation in the human genome that affects a single amino acid. And that amino acid is not deleted, but it's replaced with a different one. And just that one little change in the primary structure creates hemoglobin molecule that causes lots of problems and causes something called sickle cell anemia. So it depends. It depends on the location of the amino acid, but it is very possible that it can affect all the levels. Okay. Okay. Why don't we talk about DNA and RNA, because we haven't talked about that yet, right? You feel okay about that? Okay, I'm going to assume that's a yes. Let's erase this. Okay, DNA and RNA. And then this is the last thing I'll do. So DNA, RNA. All right. What's Chargraff's rule? I spelled that wrong.
It's the rule where adenine and thymine go together, and then cytosine and guanine go together, and then the percentages are supposed to be the same for each of them.
That is it. That is exactly right. So with a double-stranded DNA, every time you have an adenine on one strand, it will be bound to a thymine on the other strand, right? And by the way, that right there is a hydrogen bond. So another place where you'll find a hydrogen bond, which is not like in water. This is actually within the molecule. but those weak bonds are actually really important to the function of the DNA. But yes, Chargaff's rule says that A always goes with T, and C is always going to go with, sorry, C is always going to go with G, and so forth, forming the ladder, right? So in a double-stranded piece of DNA, if you have, for example, if you have a sample of DNA and 30% is adenine, how much is going to be thymine?
30%.
30%, right. Okay. Oops, 30%, that's it. What about, can you tell what the guanine and cytosine are without being given their percentages? If this is already in the sample.
Yes, they'll both be 20.
Beautiful, you've got it. Okay, because it has to add up to 100, right? And they have to be the same. So you must have a situation where you've got 20% guanine and 20% cytosine, right? Good, so Chargaff's rules work like that. So that is what you would look like if you had double-stranded DNA. In RNA is single-stranded, right? RNA is single-stranded, usually a lot shorter, right? So are you going to have Chargaff's rules involved in RNA?
No.
No, because there's no complementary pairing in RNA. There's just a single strand of, and by the way, it's going to be uracil, right? Of these bases, right? So right now, you know, what if it looked like that? It's going to be very random because it's single-stranded and there's no complementary strand opposite, right? There's nothing opposite forming the other side of the ladder for an RNA molecule. It's single-stranded. So you're not going to have the percentages that match up with Chargaff's rule. Okay, I just want you to keep that in mind. All right, what are some other differences between DNA and RNA? Details. This is the last thing we'll do. Yes, okay, so we've got uracil over here. Uracil instead of thymine. Very good, that's one, definitely. You think of anything else? Anything
The
other than...
The sugar has an extra
Yes.
oxygen, so it's ribose instead of deoxyribose.
Yes, okay, so over here we have deoxyribose, which has one less oxygen than the RNAs, which is just regular old ribose, right? And ribose has that oxygen in the ring that if you look in the very same place on the deoxyribose, the oxygen is missing. So that's how another way you can tell. And then your RNA is always single-stranded, generally single-stranded, much shorter also, and has different function. RNA is for transferring information, whereas DNA is usually double-stranded. There are other types of DNA that exist in nature, but has deoxyribose in it and is going to have thymine in it, right? Okay. How are you guys feeling? That's 40 minutes. So what do you think? Feeling good? All right. I don't know. I think some of you have still not done the progress check. I recommend that you do. The progress check is think of it as an ed puzzle. Okay, you just need to do it. I'm not grading it for accuracy. It's in AP Classroom, and the questions in it are going to be very similar to the ones that are going to be on the real test. So it's worth your while to do it. It's a very good way to study, and you don't want to get a 0 out of 10 when all you have to do is try it and get a 10 out of 10, no matter what score you get. Okay, so definitely do that. And I'll be watching for any any anxious questions tonight and tomorrow night if you have them. Okay. Okay. I think I'm going to end the session. Does it show? Oh, the progress. I don't know. You'll have to tell me. It's created for us. Oh, good. I'm glad to hear that. I think it does give you feedback. Yeah. And I think it would be really good practice. It is open notes. I would be aware of that though. Don't let that make you think that it's okay not to study, because it's going to be much easier on you if you really know your stuff, because the questions are application questions. Some of your notes will help identify what kind of molecules and things like that, but they're not going to help if they're asking you, if they're giving you a situation and you have to use your knowledge to do it. Okay, so make sure that you study, and you can have your notes out. That's fine. And I'll see you at the test. Okay?
Thank you.
Thank you. You're very welcome. It was great to see you. There's a lot of you here. That's encouraging. That's encouraging. Okay.